All right, well, welcome back. This morning, we're going to be talking in Module 2.1 about principles of modeling. And some people may say that you want to get to building code right away, and I understand that. But historically, we have done some surveying of people who started using a package like CompuCell or a competing package. And we tried to do something which is not so easy to do, which is to ask people who tried and stopped using software packages of this kind why they stopped. And when we did that, we expected people would say things like, oh, CompuCell crashed, or we didn't find CompuCell very useful. But in fact, what we found was that most people who didn't use this kind of modeling stopped using it because not technical reasons, but rather because they were having trouble thinking about how to build meaningful models. And so this is going to be a lecture and some exercises that are going to focus on thinking about modeling rather than building computer models. And I realize that that may be a little bit different from what you're expecting, and some of you may have had this experience already. If you're an experienced modeler, perhaps you could chime in and tell us about this. But I think that in general, I hope that this will be of interest to you going forward. I also have to remind you, as usual, that this is live streamed and recorded for future use, and you should be aware of that as we go forward. So the learning objective is really to try to help you think about modeling in a way that may improve the probability of success. And I would say the single most important thing is to start thinking carefully about what it is you're trying to achieve with a model before you start writing computer code. If you start writing computer code and the conceptualization of the model you're implementing is not useful, no matter how much code you write, the code is never going to fix the issues that you had in thinking about the problem in the first place. And because of the complexity of biology, taking the enormous complexity of real biology and trying to reduce it to things that are simple enough that they can be modeled and are still meaningful is not trivial to do. And I'm not going to have time today to go over all of the things that I would like to be able to talk about. Historically, sometimes this would be, when we did these meetings in person, 4 or 5 hours of discussion of this kind. It's a little harder online with the limited format. And I'm always a bit surprised that there isn't more literature and discussion of these kinds of things. We made reference to that PLOS paper by those Canadians about computational modeling, but that's a very different kind of computational modeling from the one we're doing here. And so we're going to talk a little bit about biology at a very high level and some of the questions and things you might want to think about. There are going to be additional slides that are going to be hidden, which you could look at in the deck if you want more information. We are going to go over just a couple of things in detail and maybe give you a style of thinking about problems. And then we're going to talk about practice of modeling. This may seem a little bit formal to begin with if you haven't modeled before, but it's a way of thinking about how you build and use models. And again, that kind of abstraction winds up being quite important to build models that are useful. And then we're going to talk about the workflow of building a model, because there are formal stages in model development. Whether you do them in your head or on a piece of paper on a computer, you have to go through these steps to build a model. And it's better from the point of view of building models that work, that you do it explicitly rather than implicitly or tacitly. It may seem like you want to run as a horse and that this is a bit holding you back. But after many years of experience, I can tell you that taking the time to design things at the beginning actually saves time in the end. However much it can feel that you're taking too much time at the beginning. And then we'll do a couple of little exercises. We're going to go back to the self-sorting example that Hayden went over yesterday. Think about it from this point of view. Yesterday we jumped right into the modeling. And then there'll be essentially a short exercise and then a homework problem, which is for those of you who have a specific biological system that you're working on, try to think about it in this way. That'll help you start if you're going to, for example, do a hackathon project. If you're not committed to a particular biological system, I'd ask you to pick any biological problem that's appropriate of the kind we talked about yesterday as examples of what CompuCell can do, and try to do this kind of analysis. A full analysis of this kind is quite time consuming, and so I'm not expecting people to do all of it right now. But if you were going to do, say, a PhD thesis using modeling, this would be essential, whether you use CompuCell or any kind of modeling package. We have a couple of worksheets today. I'd ask people to download those. You can use the tiny URL CC3D day 1 module 1, should say day 2 module 1, and that would be typo, or this QR code, or if you go into the student materials folder and you look in the directory, the slides, and then look for day 2, you'll find a directory which has this material in it. And so if people are having trouble finding that, I'd ask people to go back and let people know so we have it. What you'll find in there is a Google Doc, which you can download, make a copy locally so you can edit it. If you want to turn it into a Word document, that's fine. So you have that worksheet, and then there are going to be 2 examples where people have essentially completed a version of that worksheet for particular biological problems that you could use to see how those things can be filled out. So again, the main secret to successful modeling is that you don't start writing computer code or model specifications until you've really thought out and written out the aims of your model and its architecture and elements. That may seem obvious, but having worked with people for 30 years in this field, I can assure you that people often don't do that. And when they don't do it, they often wind up building models either that don't work or that give results that aren't fundamentally useful from the point of view of understanding biology in a scientific way. And so the more you spend time early in your problem defining the problem, the more likely you are to be able to build something that's going to be valuable later on. And I want to say something, which is you're going to hear some of the terminology I'll use may seem a little bit unusual. There is a whole language and jargon for how you describe these kinds of things. I try to follow the standards in this area. If you're interested, there are Wikipedia articles on many of these topics, which I recommend. There's a whole Stanford philosophy wiki, which is almost as big as Wikipedia, which discusses these issues in great detail for those of you who are really interested in these problems. And for those of you who have a historical bent, I might say that the issues of how you conceptualize models were first really discussed in a serious way already in the 18th century. If you go back and look in the Diderot d'Alembert Encyclopedia, which was written around the time of the French Revolution, they're already discussing exactly these issues. Of course, they weren't doing computing, and the terminology they use is a little bit different, but you'll hear the word conceptual model a lot, and that was already defined and discussed in the 18th century. And we're still learning how to use those ideas effectively. So some high-level things that we're going to come back to, which is the things that you need to do most when you're going to build biological models is to start out by defining high-level biological questions that you can answer. And it's not always easy to do that. When people start modeling from nothing, especially experimental biologists or clinicians, modeling seems to be something rather alien. It's a new technique, just the way there's a new imaging technique or a new omics technique. And there's a tendency, there's the famous line that sufficiently advanced alien technology is indistinguishable from magic. So there's often an assumption either that models can do nothing or that models are magic and can do anything. Models, in fact, are a tool. Building working and useful models is a lot of work. You have to know a lot. And sometimes an experimentalist will say, if I have to know that much already from experiment, it's not worth building a model. I already know what I need. That's okay, because the exercise of building the model can reveal the experiments you need to do. But building models is a lot of work. And so it's important to think about what you want to get out of them. It's really important to be specific about the biological processes, both on the input side and the output side of your model. What is it you have to know to build the model? What is it you want to predict? And in particular, how does it correspond to experimentally measurable observations? There's nothing wrong with building what a physicist would call a toy model to explore concepts. But ultimately, from an engineering point of view or medical point of view, experimental biological point of view, if you can't tie the model back to things that you can measure as inputs and the outputs, which can be measured for validation, the model isn't giving you really useful information. Key point, which I'll repeat many times, is that it's critical to think about what information a cell has available to it. The active agents in biology at the scales we're working with are always going to be cells. They're the only things that are alive and doing things as agents. And you have to think about what information a cell can have. It's critical. Cells don't have a roadmap. A cell in your body doesn't know that it's in the liver, in the lung, anywhere else. It doesn't know what's far away from it. It has its information about its internal state and its information about its local environment. And actually, if you're an engineer and you try to design self-constructing systems, self-repairing systems based only on local information, is quite challenging to do. Most engineering is based on blueprints where you have global information. Biology, you don't have global information, and that locality of information is what gives rise to the heterogeneity and robustness of biology, also to the failures in biology. So we have to think about the locality critically. If you need to put in something that's not a possible, teleportation, long-distance communication, and those things don't exist in the biological problem, then there's something fundamentally wrong with the way you've conceptualized your problem. Another way to analyze that, and my colleague Paul Macklin has just published a paper in Cell on how to formalize that description, is to think about signals. What sends a signal? How are signals transmitted in space and time? What receives the signal? How do those receivers change their behaviors in response to the signal? If you know the answers to that at multiple scales throughout your problem, fundamentally you can probably build a meaningful biological model. If you can't answer those questions, you're either going to have hypotheses and alternatives for those things, or fundamentally you can't build a meaningful biological model. Biology is full of feedback loops. It's full of feedback within cells, between cells in their environment, because cells create the environment that they respond to, between organ systems, and of course, we have feedback with the environment that we create outside of ourselves as organisms. It's really critical to think about what feedback is. If you don't have feedback in your system, then classic omics and bioinformatics techniques work quite well. Modern machine learning initially was primarily inference, which doesn't handle feedback. Contemporary LLMs, which are recursive, do handle feedback. And one of the reasons LLMs are so powerful is because they've incorporated feedback into their architectures. We're only going to be able to talk about a few examples of biological processes and questions today. And this is more to give you a flavor for how to begin to address the questions you're going to need to solve if you want to build meaningful biological models. I want to start out by thinking from the top down, from outputs of biological processes. This is a paper by the great developmental biologist Stuart Newman, and he's talking about different mechanisms that occur in embryogenesis and also in wound healing, and to some extent in cancer, where you have reorganization of tissues, and there are particular patterns of reorganization. Epibole, which is spread of an epithelial layer. Invagination, where you get pocketing of an epithelial layer, folding of an epithelial layer, often leading to pinch-off and formation of tubes or vesicles. Involution, folding of epithelial layers. Ingression, where cells leave an epithelial layer and move somewhere else. Intercalation, where cells in multiple layers thin out into a single layer. And you can read some of the others as well. All of these involve cell-level processes which give rise to macroscopic reorganization of the tissue. And if you look at the human body, we're mainly built up of layered sheets of things. We're primarily composed of epithelia with a little bit of connective tissue. And so those epithelia are rolled up into sheets or layered to build three-dimensional structures. But our fundamental architecture is based around sheets, two-dimensional structures. Then you could begin to ask the question, what are the mechanisms, both in terms of forces and mechanics, that could give rise to these macroscopic behaviors? Once you've done that, you have to ask the question, what are the cells doing and how are they controlled? Cells can move. They can apply forces and respond to forces. And you'll see some repetition of this coming up. Cells can polarize. The basic CompuCell cells are isotropic. They don't include directionality. But many cells are polarized. A cell in an epithelium has an apical and a basal polarity. A neuron has a dendritic region and an axonal region, so it has a direction. Many cells in epithelia also have a polarity in the direction perpendicular to their apical basal direction, which is called platter polarity. And Giulio, in week 2, will show you how to model platter polarity in development. Cells, of course, have a metabolism. They have to take in nutrients and get rid of waste products. They take in oxygen and glucose, other key substrates. They convert them into energy and into the components that allow them to build additional cells and to maintain themselves. There's growth, so the synthesis of new material in the cells. We often mix growth and cell cycling in our models, but biologically growth and cell cycle are separate. You can have cell division without growth of the cell, for example, in early embryonic cleavage, and you can have also, I said growth without cell cycling. I meant exactly the opposite. I'm sorry. You can have cell cycling without growth, as in early embryonic cleavage, where the initial egg divides and divides and divides without a lot of synthesis except of DNA. Or you could have hypertrophy, where you have growth of cells without cell cycle. Cell cycle includes a lot of different steps. Gilberto will talk about that. We don't model those typically in a lot of detail, but you can add those models, and it would be nice perhaps to extend CompuCell to include those by default in cells. If somebody wanted to do that for us, we'd be happy to. Cells die. Cell death is a complex process that plays out in time. There are different kinds of cell death. Apoptosis is the classic controlled cell death. There is more or less completely uncontrolled death, which is necrosis, where the cell basically lyses without doing much first. There are all sorts of intermediates like pyroptosis, which have different effects on the environment, what happens to the components of the cell after the cell dies. There's cell volume regulation and chemical homeostasis. There's cell differentiation. And I'm sure you can think about other things that cells can do. In our models, we're going to have to analyze what those are, which of those things are critical in our situations, and select only a very small subset of them. For example, typically we're going to assume that the cell's metabolism is running. When you're driving a car and the engine is working, you don't really care about how the spark plugs work. When the car is having a problem, then maybe you start looking into the specifics of the engine function. So you can ask questions about all of these different mechanisms, how they work, how they're controlled. If we're thinking about differentiation, proliferation, and death, we could look here at Wikipedia. Here we have a picture of a cell. We have an apoptosis box here. We have a cell proliferation box here. We have a gene regulation box here. And we have on the surface of the cell a variety of receptors, transmembrane receptors. Some of these are ones that you might recognize, integrins, G protein coupled receptors. Some of them might be less familiar. Wnt receptors here, cytokine receptors. The cytokine receptors are typically involved in stress response, immune response, often lead to cell death, controlled cell death. There are things like FAST and TRAIL here that are more cell death receptors. And the cell is always comparing its internal state and its external environment to make decisions about, do I stay where I am and survive, survival factors? Do I grow? Do I proliferate? Do I die? And if I die, how? One thing that we learned during the COVID period, which was quite interesting to me, is that a lot of the receptors, for example, for infection in cells, are coupled to cell death mechanisms. Primary way cells respond to bacterial or viral infection is by killing themselves to prevent spread of the virus or bacterium. The same receptors, which are the toll-like receptors, exist in humans and cats, but they're wired up to different cell death pathways. Ducks also have different ones. And so influenza infection or a salmonella infection in human will cause different kinds of cell death from what it would do in a cat or a duck. And that means that the way the disease plays out at the whole body level is completely different. Once you've decided what are the signals that cause your cell to grow, divide, and die, you need to ask how those signals interact with the internal state of the cell. If you really care about how those things change, it's not just if the level of X is bigger than this, kill the cell, then you probably need to start modeling the signaling and the gene regulation inside of the cell. In many cases, things don't happen in that way during the model and during the period that you're interested in. In that case, you can neglect a lot of this complexity of control inside of the cell. Again, remember that cells only know their internal state and local signals. They don't have information that is long distance. And so when you look at those receptors on the surface of the cell, it gives you a sense of what information is available to the cell. And so understanding that is a critical part of building models. Differentiation is still actually rather mysterious. What is differentiation as a biological process is still not fully understood. When you model differentiation, you'll find that there are particular ways of modeling differentiation. They're classic ones. Those differentiation models cannot actually explain how differentiation works. You may have heard of Waddington landscapes and things like that. They really fundamentally don't make any sense. We accept them because they're approximations to reality that give us some useful information. But, in fact, we don't know a lot about differentiation. Gene methylation, acetylation of histones, these kinds of things are critical. We don't really know how those are controlled. But we're going to neglect that. When we don't know things, we have to plow ahead anyway. What are the types of cells that are important in your system? What signals cause cells to change their differentiation state? Is that due to some external signal, due to cell-cell contacts, due to cell history, due to cell extracellular matrix contacts? Those all are important in differentiation and in death and in proliferation. There are things like contact inhibition of proliferation are very common. There's cell death due to integrins contacting the wrong substrate, very common. There's cell proliferation due to cell contact with certain external substrates, common. There's inhibition of contact, cell proliferation due to certain contacts, also common. What are the thresholds for these transitions? How do the signals interact if they're competing signals? What are the rates or probabilities of the transitions? Do I need a micromodel or not? Cells interact with each other in a variety of ways, and you'll see a little bit of interaction with environment here too. Cells stick to each other. There's a beautiful book by Forgox and Newman on developmental biology from a biophysical perspective, and the first sentence is that the most important thing cells do is stick to each other, because if they didn't, we'd all be unicellular organisms. They also stick to the extracellular environment. They use different molecular species to do that. The molecules that make cells stick together, we tend to model simply as adhesion. However, they also send signals to the cells. And sometimes it's very important to know what those signals are. Cells can create forces, directional forces, non-directional forces. They can send out filopodia, for example, that can pull over long distances. That's actually rather hard to model. And those forces are generated in controlled ways. CompuCell is pretty good at simulating some of those, not so good at others. When we get to more advanced topics, we'll talk about that. Cells polarize. I've mentioned polarization, both primary polarization axis and planar polarity. Cells are motile. They move. They move by cytoskeletal modulation. Interestingly, most of the active forces generation is contractile rather than extensile in cells, although the net movement may be extensile. Bacteria and other unicellular organisms also have additional movement mechanisms. So if you want to study bacterial movement, you want to know whether they have type 4 pili, flagella, and so on. Cells secrete and absorb molecules in their environment. There's a lot we don't understand about the basic science of how extracellular matrix is built. And cells signal a lot through contact. And we'll do some modeling on Friday of contact signaling between cells. Adhesion. We've already talked about adhesion yesterday. How strongly do cells of one type adhere to cells of another type? If it's type dependent, it's easy to do. If it depends on the exact number of which adhesion molecule in each cell, and every cell is different, then we have something called contact adhesion flex that'll help you do that. How strongly do cells of a given type adhere to ECM of particular types? And how does that adhesion change in time? How do cells interact with their environments? I apologize for the overlay here. What chemicals do cells secrete and absorb? There are lots of questions about transport, some of which CompuCell will help you with, some of which you have to do externally. If you have chemicals, they're going to diffuse. How fast do they diffuse? If they're not diffusing, you have extracellular matrix or fluid, what are the chemical and mechanical properties? How stable are those chemicals? Almost every chemical decays, but some chemicals have lifetimes of minutes, some of years. Even your bone is replaced over a period of about 15 years in your body. There are very few cells in your body that are there the whole lifetime. Some neurons, a few other cell types. But in general, there's turnover. So timescales are critical. And one of the most important things you can do when you're quantifying things, people ask, how do I know what the values of parameters should be, is to define the timescales. Does a process take a minute, an hour, a day, a year? Even having a rough idea of how fast things happen. What's the cell cycle time? What's the doubling time of the cell volume? What's the speed with which a cell crawls? All of those timescales are things that can be observed and can be used to quantify and narrow down your models. How do cells move in response to chemical signals in their environment? That could be chemotaxis to diffusible signals, haptotaxis to substrate signals, or some combination. Typically, we're not going to be modeling fluid flow here in this part of the course, but cells do respond to pressure and flow in their environments, especially in epithelia, if you have the lining of the urethral tube, for example, or a kidney. How do cells change in response to all of these signals? Those are questions you might want to ask. And that thing that was pasted in here, if you want information on numbers, a lot of things you'll have to trawl the literature for now, or maybe ask ChatGPT or some other LLM for. Don't always trust those answers until you double check them. But there is a beautiful database that's been prepared for many, many years by Harvard called BioNumbers. It's sometimes frustrating because the numbers you want aren't there. Sometimes it's great. You say, how big is a typical hepatocyte? It'll tell you probably. And I strongly encourage people to look at the BioNumbers database if you want to know how many base pairs there are in human DNA, things like that. Okay, so that's a lightning introduction to biology and some of the questions one might ask. There's a question in the chat about modeling bacteria and single-celled organisms. There are plenty of models of bacteria that have been done inside of CompuCell. You're not probably going to model what's going on inside the individual bacterium in a lot of detail. The number of bacteria in a lot of systems before you get meaningful spatial organization is very large. Often you need to model hundreds of thousands or millions of bacteria. And in that case, perhaps CompuCell isn't the best choice because we can model 10,000 cells, 100,000 cells, but modeling 1 million cells is a bit of a stretch for CompuCell. Our friends at the University of Connecticut, around Agmon and his old colleagues at Stanford, Marcus Colbert, have been trying to model all of the biological processes inside a single E. coli. They have a model with 19,000 experimental parameters in it and about 10,000 distinct submodels of biological processes for one E. coli. Probably you don't want to be doing that as a regular basis, but it's impressive that they can do it, and they can model individual E. coli, their replication, their resistance to antibiotics, and the formation of biofilms, for example, using that method. By the way, CompuCell works inside of that framework. So if you wanted to couple CompuCell models to those kinds of models, it's possible. Iran Agmon has done that. We can talk about that later in the class. There are simulations, actually, I showed the other day about response to pressure and flow. Those liver models that I showed did that. But the flow models themselves are not typically done in CompuCell. The one I showed did have the flow actually implemented in CompuCell. But typically you'd use another package and couple it. That's also something we could do in the advanced part of the course. Now I'm going to talk a little bit about something that may seem abstract, which is how you talk about models. So the first thing is, what is a model? A model is something that takes something called a model structure, a framework, and a set of input parameters, and returns some kind of meaningful output that can be quantified. And if you can't measure those input parameters, you have to have hypotheses about what they are. And that means you're going to have a lot of combinatorics. And if you can't compare the output metrics from your model to something experimental, then your model exists really only for the purpose of thinking about the problem, and you can't apply it. And so it's really critical that you think about what it is that you can measure both on the input side and the output side to constrain your model. Model structure is a little bit more tricky. Most classical biology talks really about model structure. Whenever you see a diagram, for example, a pathway diagram, that's a model structure. It doesn't have rates of reaction. It doesn't have amounts of chemical. It's purely a framework that describes interactions and relationships. And if I were giving a full course, I'd spend a whole lecture or two just describing what model structures are. But if you've done network modeling, you're very familiar with model structures. There are graphical representations of the kinds of models that we do with CompuCell. They tend to turn into hairballs and to me are not terribly informative, but frameworks like Morpheus or Vivarium will give you framework diagrams actually showing the model structures explicitly. They tend to get quite complicated. Steps to develop useful models. Some of these we've talked about before. What are the hypotheses you want to test? Be specific. You can't just say, I want to model cancer. Start with that. Until you make that more specific, you're not going to be able to develop a meaningful model. How would you validate or falsify these hypotheses? By looking at a time series of tissue configurations and cell states, because that's what you can measure. Maybe also things like blood chemistry if you're doing something in vivo. How would you quantify those observations? Sometimes qualitative information or semi-quantitative information. Biologists love differential experiments and semi-quantitative information. If I do this, this goes up. If I do this, this goes down. If I do this, this takes longer. Those are semi-quantitative results. Quantitative results, if I do this, I get 2 cells. If I do this, the cell cycle time is 3 minutes. What are the agents or objects in your system? If they have types, which types do you need? You're always going to make simplifications. Are there critical chemical fields in the system? What do those agents or objects do? That's what you're going to model. You're not going to model everything that they do, just the way when you're driving the car, you don't care about how the engine functions in detail. What are their key properties and behaviors? What are the processes? What are their key attributes? Pay particular attention to interactions and signals, as I talked about. What are the key mechanisms that determine how things change in time? It's called dynamics. Continuum changes in time. For example, cell crawling, a cell pulling on something. What determines those speeds and time scales? How would you describe them mathematically? Are the behaviors of cells basically steady during the time you're looking at? For example, in the simulations yesterday, we said that the cells had an adhesivity and a volume. They didn't change during the simulation. The configuration of the simulation changed, but the individual cells didn't change their properties. That's very useful. It makes things a lot easier. If you have to have control, then you have to decide what level of detail you want to define that control with. Very important issue, somebody brought it up yesterday when they were doing cell sorting simulation, is that the real world doesn't have edges. Our simulations always have a boundary, a box. What happens at the edge of the box? What happens at the beginning of the simulation? What's the initial configuration of the cells? What's the state of those cells? What happens at the boundary? Do cells disappear when they cross the boundary? Do they stick to the boundary? Are they repelled by the boundary? We often use periodic boundary conditions, which never happen in the real world, but are a way of avoiding having hard boundaries at the edge of a simulation. That happens with all simulations. Boundary conditions are critical, and we don't think about them as much, perhaps, as we should. Can we, in fact, make some reasonable cuts in the possibilities of our model space just by thinking about the system? Be very Aristotelian. Aristotle didn't believe in experiments very much. He thought you could do everything by thinking. We can try that. There are a lot of things that you know should or shouldn't happen, that can't or can happen, and those can often constrain the particular kinds of experiments and the particular kinds of models you build. And then what kinds of differential experiments should have predictable differences in outcome? Again, biology experiments are usually differential. These days you're getting more absolute measurements, but typically they're differential measurements. If I do this, this changes. We're going to use a certain terminology. Objects are things with properties and states. Behaviors and interactions, processes, are things that act to change object properties and states. However, they don't actually play things out in time themselves. Dynamics are things that take object behaviors and interactions and play them out in time. If you're a physicist, the force on a cell is a process. Newton's law, or overdamped force-velocity relation, is a dynamics. Events are things that happen discretely in time. They happen in a time instant. Cell division might be an example. Cell death might be an example. Existential changes, cell differentiation would be an event. Putting a chemical into the cell environment would be an event. And initial and boundary conditions. If you cannot define objects with their properties and states, behaviors and interactions, dynamics and events, initial and boundary conditions, you can't build a model. That's not about a CompuCell model. It's any model. Conceptual model, computational model, machine learning model. You have to always have these four elements to be able to build a model that's meaningful. These are always constructive models. You don't get anything for free. You don't inherit anything. You have to do all of this by thinking about it. In our biological systems, you've seen this slide before, we typically are going to be thinking about chemical fields, cells, networks. There are certain things that we know about how chemical fields work. They have concentrations, they diffuse, they decay. Cells have behaviors like motility. Dynamic networks, gene regulation, signaling metabolic networks. There are concentrations of chemicals or amounts of chemicals. They have chemical decay. You can have chemical reactions. And you can have the interactions between these things. Cells can absorb chemicals from the environment. Networks can induce the cell to differentiate. And so those are some things to think about. I'm going to run through this a bit quickly, but here's some basic things you might want to think about. Objects, the things you're modeling. The cells in an embryo, for example. One thing to remember in a model is that the universe or environment is an object itself. And from a technical point of view, it's actually rather important to remember that. The boundary conditions come about because the universe you're modeling has boundaries that aren't there in the real world. I'm going to allow the TAs to respond to some of the questions in the chat, which are really interesting ones for the moment. I'm happy to come back to those a little bit later. Properties are the intrinsic characteristics of the object. Where the volume of a cell, where it is, its shape, for example, its orientation, its elasticity, its mass. Some of those things might be thought of as variables. Some things might be thought of as parameters. Variables are placeholders, which typically change as a result of dynamics. Parameters are fixed. Now, you could always change your idea. Something that's a parameter in one model could be a variable in another. And so point of view will change things. The ensemble of variable values and parameters is called the state of an object. We often group objects by type, although type is a fairly loose concept. If you try to understand what a species is biologically, it's a hornet's nest. If you try to understand what a cell type is, it doesn't mean that any two cells of the same type are the same. Ideally, in engineering, if you have two screws of a particular type, they're the same, really the same. In biology, you have two cells that are hepatocytes that could be very different from each other. Something else to remember is that there are different classes of object. You can have a continuous object like a chemical field, a discrete object like a cell, an abstract object like a network, which doesn't exist in space. Discrete objects are a little bit tricky because the computationally discrete objects really are discrete. You could have one or two of them. Boundaries between cells are fuzzy. You could have partial division of cells where the cells are still connected to each other. You could have syncytial organization of cells. And so actually identity but continuous discrete in real world biology is actually a tricky one. Computationally, you have to make an assignment of that. Processes. What is it that cells do? Think of processes as things that are acting at a given instant. They're the things that are like forces that are going to cause change in time. But at any instant, certain processes are active, but they're not actually changing in time because you're looking at a single instant. These are the things, behaviors are things that cells do on their own. They're autonomous. Grow, move, divide. It doesn't mean that the outside doesn't affect those things, but they're things that the cell is doing intrinsically. Interactions obligatorily require more than one object to participate. Again, you put those two together in the term process. And I apologize if the terminology is unfamiliar, but these are the standard terms for these ideas. There's a key concept which I don't have on the slide and I don't want to get into right now, but processes fundamentally do not have states. There are no parameters that are directly associated with a process. Processes read parameters from the objects that they participate in. And so you can have placeholders that say you have to know what the rate constant is for a given process, but the value of that has to be pulled from the objects that are connected to the process. And that's actually a rather important mathematical concept. We're not going to run into it probably, but for the purposes of being accurate, I need to say that. It's also true that objects and interactions may be different depending on our point of view. I can think of two masses that are coupled by a spring. I can think of that as two independent objects, a mass and a spring. I'm sorry, here I said two object types, mass and spring. So I have two masses and a spring connecting them as the spring is an object, or the spring could be an interaction, a process between the two. So again, one of the problems we face with these complex models is that different points of view will have different model descriptions, different model structures. Boundary conditions. Always we have to think about initial conditions. What are all the objects in our simulation at the beginning, or our system at the beginning, and what are their states? We have to assign values to all those things. At the edge of whatever the world is that we're looking at, what are the constraints? Dynamics. Typically for forces, we have either Newton's law in the macroscopic world, or we have overdamped force-velocity relation. For things like diffusion, we typically will have diffusion equation as a dynamics. For chemical reactions signaling metabolic networks, we'll typically use rate laws, first order ordinary differential equations, although in fact those are very rough approximations to what's really happening at the molecular level. But they describe how states change in time given the processes that are active and the state. Events, again, are discrete time occurrences. They can also be things that are imposed from the outside. The state of the model at a given time after the initial condition is a result or prediction of the model elements. That's what you get out of the simulation, or for that matter, the conceptual model. Workflow models. There is, in fact, in your mind, when you build models, whether formally or informally, a series of things that you have to do to build a meaningful model. And there are a bunch of steps here. I'm not going to be able to go into a lot of detail about all of these steps, but you're always going to start with biological observations, and eventually you're going to wind up with a simulation, and then you may apply that. And it's important that if you short-circuit this process, and we always do a little bit of short-circuiting, but the more you short-circuit this process, the more likely you are to wind up with models that are not meaningful, that can't be fixed. To begin with, we have to have some set of biological observations. We have to have some idea of what it is we're trying to predict. There must be some phenomena that attracted our interest. And we're going to typically have a tacit mental model of what's going on. We've read things, we've seen pictures. We want to ask immediately what the questions are. What are the inputs, objects, processes, What could we measure at the molecular level, for example? And which ones do we think are most significant? And that filtering may be done by literature searches. It may be done by an LLM these days. It may be done by us just thinking about the problem. A lot of times you draw things. Biologists are very good at drawing sketches and schematics. Those are beautiful ways of creating mental or conceptual models of biology. Once we've identified behaviors we think are significant, how do we think they're controlled? Again, signaling is critical. Once we've defined this general area, we want to refine it. What's our subject domain? Can we define specific quantitative questions? If we can't make quantitative questions, it's going to be hard to build meaningful models. How would we measure these things? What are the metrics? How do we quantify the input and output states of our problems? Some things that we care about may not be directly measurable, in which case we have to think about how we could back them out of the situation. What are our governing hypotheses, both for model structure, like network architecture, and for parameter values? Can we constrain them? Cells don't have a cycle time of a minute. Cells don't usually have a cycle time of a decade. Typical cell cycle times would be, if for human cells, would be a day or two. Can we constrain the parameters? What are the parameters we're going to care about? Once we've got that down, we can begin to ask those kind of questions more formally. What are reasonable values for the parameters in our problem? What are reasonable results? We shouldn't have an infinite number of cells at a finite amount of time. Cells shouldn't fall apart or disappear spontaneously. Random movement should be random, not directional. If we push harder on a cell, it should move faster. There are a lot of simple sanity checks that we can do. Very often, if we're not careful, we build simulations which don't obey those sanity checks. And once we've done that, we build a conceptual model. Conceptual models don't have quantitative elements. They talk about the objects, the processes, the boundary conditions, the dynamics, their relationships to each other. These are all the things you would get in diagrams. Wikipedia has a good article on conceptual models if you're interested. Once you have that conceptual model, or what could be called a conceptual model template, then you can begin to make it quantitative. And you do that by defining the parameters that affect particular relationships. For example, if chemical A turns into chemical B and vice versa, we say that there is some rate, VF, which depends on the concentration of A in some set of parameters, at which A turns into B and vice versa. I've explicitly not given a value for those parameters or a functional form for that relationship. That's the quantitative model template. By the way, all of this should be agreed upon by anybody, whatever computational methods you're using. At this point, you're not involving computation. So anyone who looks at this problem should be able to make the same decision that you've made. Then you define a mathematical model. You say, okay, now I have a rate law for A going to B. I'm going to say it's mass action. Rate constant Ka times A is the rate at which A becomes B. You notice I've taken a framework, a diagram, and I've decorated that arrow in that diagram with a rate law. That is typically what we do when we build models. We're going to use a Hill function. We're going to assume that there's a uniform probability in time of something happening. We're going to assume that something happens a fixed amount of time after something else happened. We're going to assume that there's a linear force-velocity relationship. These are all kinds of mathematical models. Again, we haven't made any computing decisions at all when we do that. This could be implemented with a continuum ODE approach, could be implemented with a Gillespie approach or something else. Nevertheless, the basic mathematics of the interconversion rates of the chemical reaction would be the same. Then you're going to choose methodologies and pick the values, initial values for the methodology-independent parameters. Those rate constants, for example, in that previous picture, we're methodology independent. The rate of conversion of A to B doesn't care whether you're using a Gillespie or an ODE solver. Again, those method-independent constants are things you should be able to infer from the real world or from thinking about the problem. Once you get methodology-dependent parameters, however, those are things that are specific. If you are modeling a cell on a lattice in a cellular Potts model, GGH model, then you're going to have to say how many lattice sites are in the cell. If you're using a center model approach, there's going to be an interaction radius, and the cell doesn't actually have a volume at all. If you're doing a subcellular element model, you might have to ask how many components am I going to put into my cell. And so there are going to be a whole bunch of methodology-dependent descriptors that are going to be specific to the methodology you use. And you want to keep this as late as possible in your problem, because the moment you start using methodology-dependent descriptors, your problem isn't generalizable. If I've written it for CompuCell, it's not going to work with PhysiCell anymore, because you've got all of these, what in machine learning would be called hyperparameters, that are specific to the way you're solving the problem. Those don't reflect the real underlying physical and biological reality of the problem. They're extra because of the particular way we're solving it. And then you solve the thing as a simulation. And you'll notice I only have one slide on implementing as a simulation. This whole course is on simulation. But in fact, the hard part is the first stages. How do you take the initial biology and turn it into something that can be turned into a meaningful model? Building models and applying them is an iterative process. And there really are two cycles here. On the left, we have model construction, and that's what I've been focusing on in my discussion so far. And on the right, we have the application of the model to generate knowledge. They're related to each other because as we apply the model, we discover their deficiencies, and we have to refine the model. And so there's also a cycle where model refinement happens from using the model and then coming back and refining it. A critical thing that we get a lot of times, and I've mentioned before, is we build the model. Sometimes when we're building it, sometimes when we're using it, we discover that there are experiments we need to do to nail down, is X actually interacting with Y? That would be about model structure, or how fast is X interacting with Y, which should be about model parameterization. Very often, we don't know what experiments we really need to do until we built the model and worked with it and realized what its deficiencies are. And so model refinement is a continuous process. It's fine to start out with a model that doesn't work very well, as long as we have a strategy for making that model more realistic or more meaningful, because realism maybe is a little bit of a long-term goal, too hard. Predictive would be a way of maybe better word. To begin with, model construction is experiment knowledge to simulation. That's the workflow we've outlined. Once you have an initial simulation, you have to validate it and refine it. If you're going to use it in a medical context, you have to have a lot of information about how reliable the model is, when it works, when it doesn't work, how accurate it is. What's called uncertainty quantification and validation. Models are never done. You're always going to be refining them. Each time we use a model, we'd like to be able to understand what its deficiencies are and use those deficiencies to improve it. When we apply the model, we'd like to be able to design experiments from the simulation, test hypotheses, control or design the systems we care about. Find a new drug, design a patient-specific dosing schedule, design a tissue engineering platform. Knowledge capture, can we increase our understanding by understanding how models work and fail? Reproducibility is a critical component of modeling, which is something that is sorely lacking in the kinds of modeling we do here. Ideally, if we do a model in CompuCell and a model in PhysiCell of the same biological process, they should give the same results. Ideally, the model specification would be something we could carry from CompuCell to PhysiCell, run the same thing twice. In practice, we can't do that at the moment. We have OpenVT is devoted to trying to make that possible. If people are interested in that aspect of modeling, I'd love to talk to you. So after hearing me talk for an hour and 10 minutes, an hour and 5 minutes, let's do a little model. We're going to do this. I'm going to run through this. I'm going to ask, the frame is as an exercise. I'd ask you to download that worksheet. And you can use that worksheet as a reference here, but we're going to run through this example. And we're going to go back to that cell sorting example that we did yesterday as a simulation yesterday afternoon. And I want to come up with a few of these things. I'm not going to be able to go through everything in detail, but I hope that having seen it once, asking these questions will be reasonable. Where did that problem come from? That cell sorting problem came about because there was an observation that during development, initially mixed tissues separated to form layered structure. And the classic example of that was actually in the eye, chicken eye. And the reason that they used the chicken eye was that when those experiments were done, we didn't have very good fluorescent dyes, especially for looking at cells that were alive. And the black pigmented retinal cells in the retina were easy to observe because they were black. And these are actually experiments using pigmented neural retinal cells I did in my lab many, many years ago. Jose Mombach, who's now a professor in Brazil, and my graduate student did these experiments. We've taken pigmented neural retinal cells, it's transparent. Pigmented neural retinal cells, which are black, and neural retinal cells. I'm going to say that right the third time. We're taking pigmented retinal cells, which are black, neural retinal cells, which are transparent, mixing them in a random aggregate, and you see they sort out. The black cells go to the middle, the transparent cells go to the surface. There's a huge literature on this, mainly tracing back to Malcolm Steinberg. Goes back even before his work in the 70s. He lived a long life at Princeton University, and he developed a series of experiments to try to understand why this sorting was occurred. And one of the things that he did was a series of experiments where he took cells from different tissues and found that they made a consistent engulfment of each other. And you could measure the actual surface tension, the forces between the cells. And he found that the more cohesive cell type was always engulfed by the less cohesive cell type. And that observation led him to say that cells of different types have a contact per unit area, contact energy, which we have in our simulations, and that the pattern rearranges to minimize the total contact energy or maximize the adhesion between the cells. This is now biological observations, initial questions, and hypotheses. Now, let's just take a few minutes and ask you to think not about computing, but just about what would the key objects be in this simulation or in this model. Let's not say simulation, let's in this biological reality, because we're talking about conceptual models. What are the key behaviors? What are the key interactions? How might you determine the dynamics? Notice that Steinberg's hypothesis says that the final configuration depends on the contact energies. It doesn't say about how they get there from the initial configuration to the final configuration. It says nothing about dynamics. What would the initial conditions be? And what would be something you could change in an experiment that would change the outcome in predictable ways, depending on your hypothesis? And I'd like to give people now a few minutes. I'll give you, 5 minutes here to think that over. You can write it in that spreadsheet that I asked you to download, or you can just put it in a Word document, in the Google Doc. And I'd like people to type in the chat some of their answers. We're not going to go through this in a lot of detail, but there's no real right or wrong here. The goal is to think about what we've just seen and ask the question, if I were going to start describing this biologically, there's no modeling here, purely biological description. I've seen an experiment. Experiment gave me some results. I'm going to try to understand that experiment. How do I describe biology? Okay. Is that clear as a little exercise? So we'll give people 5 minutes to do that. And people are free to start typing things into the chat at any time. You don't have to have gone through the whole exercise. You could say, I think these are some objects that are important, or these are some processes that are important, or these are some dynamics that are important. And I see people asking questions and putting ideas out in the chat. I'm going to keep my mouth shut for the moment and let people, TAs and other people in the class, can help each other and respond to that. This is really an exercise that's most useful if you think about it together, not have me tell you what I think the answer is, although I will give you an answer that I think, and you can agree with it or disagree with it afterwards. I also want to say that I know that 5 minutes isn't really enough to do this exercise completely, but I want people to give it a bit of a shot, and then you can go perhaps and work on it more at home if you're interested. And there are also going to be a lot of slides that I'm not going to show that are in the deck that'll give you more information as well. Okay, people are putting great ideas into the chat. I would encourage you to keep thinking about it. I would start out by making a comment that from the Steinberg's work, cells have a consistent hierarchy of contact energies per unit contact area. That adhesivity depends actually on the number of cell adhesion molecules in the cell. We've had people talk about cells doing random fluctuation. Something we didn't really talk about here, but in these experiments, if you looked in detail, the cells are not growing or dividing or dying during that period. During this period of about 3 or 4 days, you don't see much cell growth or division. And we see that less adhesive cell types engulf more. So we're going to assume that cells contact adhesion. By the way, these days we know that differential contractility, active contractility of cell membranes is quite important in this process. Cells can also send out filopodia that pull over longer distances. Cells could send out chemical signals and move by chemotaxis. And one of the things we did early on was actually compare what we would expect in the dynamics. Chemotaxis sorting dynamics are different from adhesion sorting dynamics. Adhesion sorting dynamics I'll come back to in a second. And pulling of cells on each other by filopodia is also possible. And I should say that actually all of these things do happen in different biological contexts. So one of the key questions then is, how does the outcome rate of change of the pattern depend on the contact energies? And this was a question, can differential adhesion and random motility explain qualitative and quantitative observations of sorting? So we're going to start out with 2 cell types. We're going to call them dark and light because that's what we see here. And we're going to have medium thing around the cells. And that's our objects. We say the objects don't shrink, grow, divide, or die. We're going to say that there is some kind of membrane fluctuation. We're not adding or taking out medium. These cells aren't polarized. They're symmetrical as far as we can see. All of the dark cells seem to be about the same. All the light cells seem to be about the same. Medium doesn't seem to do much. Those are all experimental observations. The cells clearly stick to each other. They're not distributed in the medium, but they're not sticking so much that they can't rearrange. They don't seem to stick to medium. They seem to repel medium. We see that dark cells seem to stick more to each other than light cells to dark cells or light cells to light cells. The dynamics is random amoeboid motion of the cells, but enough that they can disconnect and reconnect. And the medium seems to be totally passive. What's our initial condition? Well, in this case, we have a blob of randomly mixed cells of the 2 types surrounded by medium. And so this is the conceptual model that we've just defined. Now, there's an interesting comment that maybe we should be thinking about the molecular basis of this. In fact, when two cells come in contact with each other, there isn't a single contact energy between them. That contact energy depends on the binding of cadherins or CAMs between the cells, and that binding changes in time. Assuming that there's a fixed contact energy per unit area between two cells of a given type is a huge oversimplification. We know in some cases that the kinetics of binding is very important. In other cases, it's not so important. Here we've assumed that it's not important, but that's a hypothesis, and it may be very well falsified. Those great, very good observations. If we want to start building a quantitative framework, we have to ask, how are we going to specify the various things we put in our model? Remember, the environment or universe is always a component. If we're in two dimensions, we have to say how big the universe is that we're in. Is it square, round? Does it have periodic boundary conditions or not? Cells have a position. They might have a mass. They certainly have a volume. They certainly have surface tension. They probably have a surface area. Some of these are parameters, things that we're going to assume are fixed during the model that we care about, or coming from something outside of our model. And some of them are going to be variables. Those are going to be things that change dynamically as in our model. Cells might be of different types. I don't know. I labeled them endoderm and mesoderm here, and medium, but we could give them other names. Now we have to think about the processes the cells participate in. The cells don't overlap. There's volume exclusion. That seems obvious in the real world. We know the physical objects don't interpenetrate, but in a simulation, you have to assert it. So there's a volume exclusion. Cells adhere to each other. Cells adhere or repel the medium. There's got to be some adhesion strength or repulsion strength associated with that. We have to have some initial condition. How big is our cluster of cells? How many cells do we have? Is the organization random or not? And so this is now building what we call a conceptual model framework that we can then fill in with the numbers we'd get either from hypotheses or from experiment. Now I want to take another minute or two to think about what are the things that we might measure about this system to quantify what it's doing. What are the outputs that we could use if I see this series of pictures? I've got a movie, either experimental movie or simulation movie. This is changing. How am I going to quantify those changes so I could compare my simulation and my experiment and say, these agree or they don't agree? If I change the adhesion between the two cells, these agree or don't agree. If I change the motility of the cells, it changes in the same way or not. Why don't people think a little bit about, give me in the chat, we'll just take 3 minutes to do this, give me some answers for things you could measure about this pattern in time or at a fixed time that could be used to compare the simulation and the real world, or for that matter, to compare two real world experiments. Something to remember, and I'm seeing people discussing really intensively in the chat, is that the pattern is random and the movement is random. So even if I start with the exact same initial condition and I had it happen in time twice, I'd get different exact positions of the cells every time. If I ran the same simulation twice with these identical initial condition with different random number seed, I'd get different results. And this is tricky. Biology is stochastic. There may be global averages. The typical volume of the tumor changes in a particular way in time. But the exact cells in the tumor would be different if I ran the same thing twice in time, if I could do such a thing. And so one problem we're going to face is that we have to come up with aggregated measurements that are robust to that stochasticity of the problem. You can't compare bitwise, say this cell should be in exactly this place, this cell should be exactly this place. There are developmental processes, by the way, in which you do get the cells in exactly the same place. For example, in the inner ear, the hair cells are exactly lined up in particular ways. There are certain cells where there's a single cell of a given type in your body, it has to be there. But most tissues, you care about the overall architecture, but where the exact cells are doesn't matter that much. People are coming up with all sorts of really interesting responses to this. Number of clusters, radius of clusters. Those are great suggestions. Correlations and how they change in time. Great suggestions. There's a comment about steady state. There's a famous comment that the only steady state is when you're dead. Even that may not really be a steady state. You do have dynamic steady states. Biology, you almost never have actually equilibrium. Homeostasis is hard. If you're sitting here and your body looks the same now and a week from now, a lot has happened to keep it the same. I didn't appreciate the complexity of homeostasis until I started working with toxicologists. Okay, these are great questions, great examples. So one thing is to think about the typical distribution of cells. And Hayden showed versions of these slides. If I have a checkerboard and if I have contact configuration, and I know what the various contact energies are, in this very simplified geometry, I can actually add up what the total contact energy is for the checkerboard. I have 8 units of light medium, 8 units of dark medium, and 24 of light dark. So I can actually derive the contact energy. If they're sorted, I have 8 units of light medium, 8 units of dark medium, 10 units of light light, 10 units of dark dark, 4 units of light dark. I can take the difference between the two. I get 20 units of light dark minus 10 of light light minus 10 of light dark. And that means that the right one has a lower energy than the left one if 2 light dark is bigger than 2 light light plus light dark. Somebody asked, how do you know what the energies have to be? This is the calculation that gives you that. If light dark plus dark dark is bigger than 2 light dark, the checkerboard has lower energy. And you'll move towards that. So now you can begin to say what the parameters are in the problem. I don't want to go into too much detail. Environment, we have to specify how big it is. And now we can begin to define the properties of the cell. It has a volume, it has a surface area, its compressibility, has an elastic modulus. The fluctuations have an amplitude. There's a dissipation in the environment. Forces are dissipated at small scales. And I don't want to walk through all of these in detail, but you can build a table that says what is the specific quantitative information I have to turn this structure of a model into something that can be computed. Initial condition, there's a lot of detail that we have to put in. The dynamics, as I mentioned here, is a stochastic movement against forces. Now I could, once I've got those list of parameters, I could say the cell volume is 125 cubic microns, cell surface area is 75 microns squared, and now the adhesivities are in piconewtons per micron squared. I could actually give numerical values to all of those things. Some of these things can be measured. The cell volume is pretty easy to measure. Adhesivity is actually really tricky to measure experimentally, it winds up. Density of cell adhesion molecules is not that bad. It's not easy, but it's not that bad. Actually measuring numerical values of adhesion is tricky because of this issue that it's because of the dynamic nature of adhesion. depending on how I make that measurement, I get very, very different estimates of the adhesion. Okay, now I can build a mathematical model. This looks like a CompuCell Cellular Potts GGH model, but I haven't said anything about being on a lattice at this moment. I've said that the cells are compressible, so there's a compression energy, a pressure energy. I've said that the cells have a contact energy between them. That would work in a center model, a vertex model, any kind of simulation. Then I have to pick my methodology and my methodology-dependent parameters, and that's what you did yesterday. And that's where that temperature came from, to describe the fluctuations of the cells as they're moving around. The blob initializer drew circles of cells, not very beautifully. The lattice type we had to pick. Clearly, the lattice type isn't something that's the real world, and people found it confusing. What is this interaction range? What's the difference between a square and a hex lattice? Those aren't things that are in the real world. Those are purely methodology-dependent hyperparameters. And then we run it, and we see that depending on the hierarchy of J's, we get different patterns. We can get mixing, engulfment, partial sorting, and those could be qualitative or semi-quantitative observations. If the experiment does the middle one and our simulation does the top one, we know we made a mistake, either in model structure or parameterization. And here's a little bit more, actually, quite a few different patterns we can get. Here's dispersal, inverse cell sorting, where the more cohesive one actually goes to the surface. Cell mixing, there are actually multiple kinds of cell mixing, incomplete sorting, and so on. And now we come back to the question that I asked before, which was the dynamics. And really, people have already been answering this question. And so I'm going to assume that we've actually done this. I'm going to skip ahead because I don't have too much time left. But we can ask the question, once we define these metrics and how they play out in time, how would we compare them? And here's how we did it in experiment. Somebody said, measure the cluster sizes. Well, we did that experiment. This is the typical diameter of a cluster versus time in this experiment. The problem with that is that there are not a lot of clusters, and so the statistics aren't great. It's also not so easy to measure. In this, it looks like a 2D image. It's a 2D image, but in fact, it's a sphere, and so we miss some clusters, at least early on. And so actually quantifying the cluster size winds up being pretty hard. But at least in this experiment, it looked like the cluster size grew essentially linearly in time at a rate that depended on the surface tension, sigma, divided by the viscosity of the tissue, eta. And this is a 3D simulation. Somebody was doing a 3D simulation yesterday. It was a very old 3D simulation, seeing the outside of the simulation, and then a section through the middle, seeing the sorting happening. One thing you'll see is you'll see clusters of light cells here that get trapped by dark cells. That trapping is more significant in 2D than 3D, but it still happens. And so you'll discover that what happens is as clusters get bigger, sorting slows down because of blockage. And so sorting actually winds up being an exponentially slow process. It's very fast and efficient over short length scales, and it's quite inefficient over long length scales. Here, what we've measured is the fractional contact length. This would be dark light contact. This would be dark dark contact, and this would be light light contact, and this would be dark dark contact here. You'll notice here time is exponential. So this is a process that's linear, is slowing down exponentially in time. And then there's a saturation, because when you get close to being fully sorted, not much happens. I'll make a comment about biological mechanism here, which is that very often biology does things using multiple mechanisms that seem to do the same thing. And cell sorting is an example of that. In early development, you'll get differentiation. Differentiation is noisy, and some cells will misdifferentiate. They'll read the signals of their environment wrong, and they'll differentiate into the state that's not the state that they should. Those can be fixed in a variety of ways. One way is through apoptosis. The cell has signals that say you're in the wrong place, and it dies because of that. That's one cleanup mechanism. Another is redifferentiation. The cell reads the signals from the environment, says, I'm in the wrong environment. I'm going to change to be the same as my neighbors. That happens. But another one is that the cells that should be together stick more. And so cells that are in the wrong place but not too far out of where they should be will reorganize and find the correct neighbors by moving around. And that's cell sorting. Very often you'll have a coarse-grained process, which gives you a rough pattern that happens rapidly over long distances, for example, through chemical signaling, long-range chemical signaling, but it's not very accurate. It's followed or at the same time as a more accurate but shorter range or slower process. And so very often in development, you'll have long-range chemical signals that give a rough pattern, get primary differentiation, and then you'll have a process like cell sorting that cleans up the mistakes from the noise in that long-range process. So coarse grain followed by fine grain mechanisms, typical behavior in development, homeostasis, and recovery. All right, so the last exercise, and we're just about on time here, we actually may end a few minutes early, is asking you to think about a problem that's of interest to you. If you're already working on a biological problem, pick that. If you're not working on a biological problem, you could pick one that you think is interesting. Problem, try to make it not too complicated. Not what's the origin of life or how do all cancers work. Could be one of the examples I showed you yesterday at the beginning of the lecture or something else, and begin to try to apply this workflow to it. If you're going to do this as a research problem, you're going to have to go through this workflow at some point. Even if you never do a computer simulation, you're only doing experiments, you still have to go through this workflow because experimental biology depends on conceptual models. And so you actually have to do exactly this, even if to define valid experiments, even if you never get near a computer. Well, these days you're always going to be near a computer when you're doing an experiment, but even if you never build a computer model. And so if you would like, maybe people could type in the chat. We'll give people 5 minutes here. Maybe what kind of biological problem you're interested in. What are the key questions you might want to answer? Think about some experimental measurements that you want to reproduce, the outputs perhaps of the signal of the experiment, and then maybe a little bit about what kind of objects you might have in the simulation or in the model. I keep saying simulation when I really should be saying model or the real world. What are some behaviors? What are some interactions? And so on. And so this framework of the questions you ask, first as biological observations and as conceptual model, then gradually making things more quantitative, slowly, and then only at the end making simulations. If you follow that, remember that everything up here, biological observations, initial hypotheses, conceptual model, quantitative model, template, everybody will agree on that. That's something that's completely shareable and reproducible. When you start making mathematical models, you're making certain kinds of decisions about things like constitutive relations. What's the force-velocity relationship? That will depend on choices you've made. Your model isn't as generalizable when you get to that stage. Once you start implementing computer simulations, you lose that generalizability. Science is about reproducibility. When you get to these lower scales, you're losing reproducibility. You have reproducibility within the modeling framework, but you're not reproducible between frameworks anymore, or between methodologies. And so the more you can refine and preserve that high-level description, the more scientific your results will be and the more useful they'll be. So why don't people just take a few minutes to type in the chat some of these things, and then we'll go with the poll and close up. Okay, there are fabulous questions here. I'm going to move on to the questionnaire. I hope people will continue to work on this. Please do fill out the questionnaire. It's important for us to get your feedback. I would love to be able to spend more time on this, but for today, that's as far as we're going to be able to go today. I hope that this was interesting for you. Please do make your suggestions. I realize that this kind of lecture is a little bit different from the things that we normally talk about in a modeling course. I'd also be interested in your feedback, whether in other courses you've had this kind of discussion of principles of modeling. Again, it applies just as much to experimental biology as to computational and mathematical biology. And so anything you can do to help us out would be much appreciated. We've been working on a paper, which we've been working on for 20 years, never published, on this topic. Anybody feels like joining us in trying to finish that paper, we'd be delighted to have your participation. I'm seeing lots of great ideas that could be turned into hackathon projects. Some of them are pretty ambitious, so how far you get in a 3-day hackathon is another question, but we find that focusing on this kind of thing for 3 days in the hackathon can be a great way of jump-starting a PhD thesis level project.